So this is chapter 14, and what we're going to be looking at, I'm not going to spend a whole lot of time on what defines a species. There are lots of different concepts of what defines a species and how we determine those sorts of things. I don't want to spend a whole lot of time because we can get bogged down. But we're going to try to focus on mostly is how do new species evolve, right? How do we get reproductive isolation, and how do we get genetic divergence between populations leading to the formation of new species? And we're going to be looking primarily at things that involve natural selection and sexual selection, causing populations to become different from one another. So this kind of ties back to a lot of the key concepts that we have discussed early in the course and are important in the speciation process. And we'll be talking about a bunch of examples of these. This, I think, is one of the coolest examples of what might be a kind of what we call an incipient or new speciation process, and it can occur relatively rapidly. So in the London Underground, anybody been to the London Underground? Ever been bitten by a mosquito down there? Okay, good. So in the London Underground, there's a kind of a, I don't like the terminology they have here, but the species in the London Underground is C. pipiens molestus, okay? And it lives in the underground. Above ground is another mosquito called Culex, difference in biology, Culex pipiens lives above ground. Which do you think is the ancestral species and which is the incipient or new species? Yeah, this is the new one, right? The London Underground has only been around for a little while, right? Culex pipien, the above ground one, has been there for some period of time. So this is believed to be the derived population from this above ground population. When these were built, water was left down in there, all sorts of things. Humans are down in there on occasion, or various kinds of mammals even. Well, not humans. Guess what kind of So these things live in really confined spaces down in here, and they bite mammals. Not only us, but guess what's the thing they bite the most of in the London Underground? Rats, okay? So there are lots of rats, you know, especially in these parts of the London Underground, the passage, the things between the sites. But obviously, there are lots of rats down in there, and they bite mammals. They do all sorts of weird things. They actually don't they don't need a blood meal to lay eggs, and they're active all year round. Okay? The above ground one lives in open spaces. It doesn't bite mammals. Its preferred hosts are birds. Okay? They need a blood meal that they need to lay their eggs, and that's what most mosquitoes need. And obviously, they're dormant in the wintertime, depending, obviously in England, it can be fairly cold. Down in here, it isn't. So, and this probably happened in the last 100 years. The divergence between these populations has become pretty dramatic. They completely eat different things. They have no blood meal, and they're active all year, okay? And this sort of rapid evolution occurred probably in the last 100 to 150 years, okay? Now, are these different species? So who says yes? Okay. Who says no? Who says I need a lot more information? Okay, yeah. So one of the things you'd want to know is, what might you want to know about these two populations? Yeah, can they interbreed, right? So one of the key things we'll focus on, and this is the biological species concept, is the distinguishing feature that we use to distinguish species is, are they capable of breeding and producing viable offspring, okay? So we'd have to probably look at that. The other question is there, we might ask ourselves, is there any potential for these two populations or subpopulations of Culex pipiens to interbreed, right? What do you think? It's hard to say. But what you'd have to know, is there any gene flow between these two populations, right? And which, maybe it goes in one direction more than the other. And I don't know the answer to this, but one of the other things you'd want to know, what is the potential for them of mating, individuals from the two different subpopulations mating with one another? It turns out there's actually very little gene flow and potential for interbreeding between these things, it turns out, okay? And I think the evidence mostly suggests that it's mostly these things from the above ground getting down into the underground, and the gene flow is mostly that way, but it's pretty rare, okay? So those are the sorts of things you'd want to know, and there are all sorts of people looking at these sorts of incipient speciation processes, right? And the key thing is you need isolation, you need divergence in traits, and then potentially complete reproductive isolation. And how do we get to that point? Okay, so here are kind of the things we're going to be looking at. What is a species and how does speciation occur? We're talking about different models of speciation, and then we'll be focusing, I'm focused primarily on what creates reproductive isolation among populations, and what are the roles of natural selection, sexual selection, and drift. All of these things, natural selection, sexual selection, and drift, can cause populations or subpopulations to become different from one another, to diverge from one another. And then we'll be looking at various kinds of models from allopatric speciation to parapatric speciation to sympatric speciation. And sympatric speciation is the least likely mode of speciation, but we'll look at some examples where it actually can kind of occur, but it tends to be fairly rare. And the reason it's harder to get limited gene flow. Okay. So if we want to go out and see things and recognize what are different species, we usually do it in a pretty simple way, at least in the initial stages. And when we look at, things, how do we sort of inherently recognize that, oh, that's a species that's different from that one, just in a general way? Yeah, phenotypically, do they look different from one another? And what are those traits that they have that distinguish them, right? We use all sorts of phenotypic markers to distinguish bird species, plant species, animal species, particular characteristics that we use to recognize. And in general, most species don't have a lot of overlap, and they're distinct and they're divergent in particular traits. So a lot of what we do is we have phenotypic traits. This could be bill length, this could be bill width, whatever it is. We can look at these things as phenotypic traits in this sort of bivariate space, and there are clusters of these things. Okay, so this might be a thing that has a short, narrow bill, right? So this could be something that has a slightly longer and a slightly wider bill, and this could be a short bill but very wide bill. Okay, so we tend to characterize individuals or populations are often highly clustered in phenotypic space. So all these individuals fall within this cloud, or that cloud. So we have kind of these three clouds. And we might be able to say we use this pattern of clustering to assign species boundaries, at least phenotypically, at least initially. That might be species 1 up in this cloud, that's species 3, and that's species 2. And this is often the first thing. This is what taxonomists do. This is what people who go in museums, they look at things and they measure all these characteristics and they try to distinguish them based on these phenotypic traits, often not just in two-dimensional space, but often in much more sophisticated means, and we're not going to worry too much about this. But here's a group of plants right here that obviously you can't see all the details, but what are some details that they differ in? Color is one thing. The thing you can't really see is kind of the flower morphology, often how long the spurs are, how long the corollas or flowers are, all these sorts of things. How some are more open than others, and all these sorts of things. But basically, and we're not going to worry about it, we can often look at these in what's called multidimensional space, right? Like not just two phenotypic traits, but we can also characterize them by 10 or 15 traits. We use all these sophisticated statistical techniques you don't need to worry about to reduce it. And basically, the basic take-home message, you still have these clusters, and I think depending on which one, I think A is associated with the green one down here, right? The B population sort of has this cluster of individuals or groups in this multidimensional space in here, and this might be C down in here, okay? So one of the things you can cluster, these are different populations, so these are 7 populations of the same species. So it looks like this cluster is separate from the data points and from these 2 other species. Okay, so one of the things this might be, the 7 populations, do they all look the same? That's one particular species of M. arborea shown within this circle, And this cluster is separate from these two other species, but is there a lot of variability among the populations? Yeah, I mean, it's hard to say how much, but some, right? Each one of these populations kind of clusters in different parts of this three-dimensional space in this cube, okay? So they're slightly different points where they occur, okay? So really one of the first things, you really need phenotypic divergence, right, between populations to eventually lead to reproductive isolation. And we can look at it in a lot of different ways. The other really important thing is, this is especially true for the biological species concept, is we often characterize things by gene flow. That is, can individuals move between subpopulations and mate and produce viable offspring, right, among clusters of these subpopulations? So let's kind of go from this point. Here you have this cluster of 4 populations. The arrows between them represent the fact that there is a lot of gene flow or movement of individuals between them, and presumably they do okay in terms of mating and those sorts of things, okay? What's the other kind of cluster we have? 4 and 6 seem to have some gene flow between them, and gene flow occurs or could occur between these populations. And then what's this one over here? Is there anything going on over here? Nope. It's not moving over to 5. 5's not moving over there. This seems to be, there's no gene flow, and this seems to be an isolated subpopulation. So we use these patterns of gene flow to assign species boundaries, okay? In that these 4 populations, 1, 2, 3, and 5, individuals move, presumably mate and breed, and no problems in terms of offspring viability between matings between individuals of 1 and 3 or 1 and 5. And so we'd assign those as species 1. These 2 there's gene flow, effective breeding between these, and then this is a completely isolated subpopulation, and we might consider that species 2. Yep. Yeah. So how do you check for gene flow in nature? So how would you do that? Yep. So one of the things you can do is you can sequence individuals from all these populations, and there are methods by which you can come up with estimates of gene flow between these subpopulations, and there are all these various computer programs and algorithms to do that sort of thing, okay? So that's kind of an indirect way. You use sort of either genetic markers or DNA sequences to measure the effective level of gene flow, and that's what most people do now. Do you think there's a more direct way to do that? Yeah, you can look at them. So what if this were a bird population? How could you assess whether an individual moves from this population to that one? Do you know what people do? They go to the bird, they go to the nest, and guess what they put around their legs? Little bands, okay? That often are unique identifiers, or they're color-coded. You know, all these individuals in this population get a blue band, right? These all get greens and those sorts of things. And then you go back sometime later and go back and see, do you find these all had blue? Do you find any blue-banded individuals over there? Why do you think people avoid doing that? You think that's really hard? It takes a long time, right? You also have the problem, you know, that you have to band a lot of individuals to get really reliable estimates of gene flow, because, you know, the fact that you can go in and recover, it depends on where you are. If you're on an island, it's a lot easier to do. But if you're dealing with something in the middle of, you know, northern Canada or something, and you're trying to find out, oh, did they come back the next breeding season to the same place or a different subpopulation? How easy do you think that is to do? It's really hard to do. So most people use these sort of genetic markers to infer the gene flow. Okay? So that's what we kind of use these. So you know, how you estimate gene flow is another matter, but lots of different ways to do it. That's what we call the biological species concept. The other thing we often use is sort of what's called the phylogenetic species concepts, and what we're going to do the next time is do a little bit on how to reconstruct trees using data. And we'll be doing some DNA sequence data. I'll give you a little homework, and you can kind of map these character state changes onto trees and things like this. But let's look, let's say we've decided this is species 1. This is species 2. So the question is, what is kind of the distinguishing feature that characterizes these two species? Is it flower color? Yeah, it's probably the flower shape, okay? Because what you can see here, flower color, right, is highly polymorphic and is not a shared derived trait of the monophyletic clades. Because what you can see here, right, is in this species you have these could be subpopulations within the species across its geographic range. This population, all these populations have orange flowers, right? But what do you see over in species 1, 2? Yeah, you also see some, it's not exactly, but I think these are supposed to be orange flowers too, okay? So that's probably not the characteristics that we would use to say that these are sort of there's some trait that arose in the common ancestor in each one of these species and is what we call the shared derived trait. What is it? What's similar? What do all these share in common versus being very different from over here? Shape, okay? So flower shape here, these flowers are all kind of the same shape. Flower shape is what we call a shared derived trait of this monophyletic clade. And what we're saying is that sort of shape arose in the common ancestor to all this species and all the subpopulations or populations within it. And it's a shared derived trait that characterize each one of these monophyletic clades. That is species 1 and species 2. And we use that to delineate or to determine the species boundaries. It's this particular trait, the flower shape, not flower color, that we use to recognize these different species, okay? So this is and a lot of people are using this now. It's called the phylogenetic species concept. And a lot of people are especially using DNA sequence data to do this exact thing. So this is the most common thing that's done. We'll do a little example that hopefully the next lecture period to show you how to do that. Okay, so those are kind of the concepts, and there's an ecological species concept in your book. Don't read about it, unless you're really interested. Okay. I had somebody come in yesterday and say, should I be reading all the material? And I said, oh, no, don't read all the material, because I can only cover about 30% at the most in each chapter, so don't read the textbook if I don't talk about it. Okay, don't waste your time, unless you're really interested. Okay. So let's now talk about process. How do you get those monophyletic clades where species 1 and species 2 are distinct? What's the process of speciation? And we're going to be talking about 3 different kinds of speciation. These are called the modes of speciation. One is called allopatric speciation, one is called parapatric speciation, and the last one we'll talk about is sympatric speciation. Is any of this, did you guys talk about any of this in biodiversity? You did? Okay, okay. So this isn't completely new. So under allopatric, we often define, and what we're going to do is we're going to kind of go through each one of these modes of speciation through time and what happens and how it then leads to genetic and phenotypic divergence and presumably reproductive isolation between the descendant populations and presumably speciation. Okay. First one is called the vicariance model. We'll talk about that. And then there's the peripheral isolate model. So what we're imagining, here's the original population. These are 2 kind of submodels within allopatric speciation. And so let's look at what's called the vicariance. Vicariance basically just means sort of splitting, okay? Complete isolation or splitting. So here's the original population. Here's the initial step of speciation in that this one continuous population has a geographic barrier that then isolates these 2 different parts of this original geographic range into 2 completely isolated subpopulations. And what do you think, what are some kind of classic examples of what can be a geographic barrier to dispersal or movement of individuals between what was once a individuals can move continuously around that entire range? Huh? Yeah, bodies of water are going to be one of the most important things, especially for things that are terrestrial and can't move across bodies of water. So that's one important barrier. Can you think of anything else? There are lots of man-made things, but we think about natural processes. What are some other natural processes that lead to isolation? Well, how about continental drift, right? The movement of continents, right? It's slow, but it isolates land masses from one another, separated by water, so that can be an important isolation event. Think of anything else? How about a mountain uplifting, right? When you think of mountain uplift, right? Mountains going up, right, can isolate if they were once continuously distributed, you know, and now you have this elevational gradient. It can often isolate populations. And there are lots of things that can lead to this geographic barrier to dispersal. The idea here is if over time, once that barrier arises, these individuals don't move between these subpopulations, what we can imagine is that they start to evolve, right, and become different from one another. And then we'll be spending some time looking at this, is you can get the evolution of isolation, of reproductive isolation, such you have a, I don't know, let's call it a blue phenotype and a green phenotype, whatever those colors mean. Those things have started to evolve phenotypic traits, presumably that are genetically based, that they become different from one another. Okay, and this is done in complete isolation. So what do you think, what are some processes that could cause that isolation leading to reproductive isolation and phenotypic divergence? These two environments might be different, right? And they favor different kinds of phenotypic or genetic traits, right? What else could happen? Yeah, it could be, yeah. So there are lots of ways the environment could vary, right? So go back to the sand mice example, right? That might be an example we could ask whether it's parapatric speciation or allopatric speciation. This gets kind of dicey sometimes and doesn't really matter. But anyway, when they colonize those sand dunes, right, they're experiencing a different environment. Predators are favoring different kinds of color phenotypes in those two different, the forested versus sand dune environments, and it leads to presumably, probably reproductive isolation, and we'd want to ask that question. Okay, so we think a lot, what we focus mostly on in evolutionary biology is the mechanisms that lead to that evolution of reproductive isolation due to allopatric is usually natural selection, right, or sexual selection. But drift could also cause it too, couldn't it? Maybe over in this one, okay? So the peripheral isolate model sometimes focuses on natural selection, sexual selection, but also thinks drift can be important. And why do you think drift is important? So here you have the original population. They're all in here, and then a little island pops up through, I don't know, some sort of uplift, you know, and an island forms, or sea levels drop, whatever it is, right? And a new island forms. And then there's dispersal to that isolated region, okay? And then there's no ongoing dispersal or gene flow, and they become isolated, and over time they become different from one another. So why do you think drift might be important here? Yeah, drift could be important here. Why? Because that's a fairly small population, right? So it could be a founder effect, right? Where just a few individuals disperse that are genetically pretty different from the other one, and they might become isolated and very different. But it also could be natural selection too. Okay. So again, the key is these populations are becoming divergent over time due to isolation and probably experiencing different kinds of environments in natural and sexual selection. Parapatric speciation is a little different in that you don't get complete isolation. So the potential for gene flow is here. What's imagined here, here's the original population, and what they're saying here is there's what's called a selective gradient across this part of the population. So the selective gradient here basically means in these kinds of parts of the environment, the blue phenotype is favored, right? So what that represents over in here, this thing right over here, blue is favored, right? What's favored over in this part of the range? Green, okay? So this is what we call parapatric speciation, where you don't have this complete isolation of these two subpopulations, but you have selection is differing across the geographic range of the population. So that blue phenotypes are favored here, greenish phenotypes are favored over here, and this is what we call, this is what's, we'll look at an example of this. This is called divergent selection, or this is called a cline. This sort of spatial gradient as you go from here over to here, this is what we call a cline. So selection varies across this cline. Blues are favored over here, greens over here, and what's kind of over here in the middle? Looks kind of mixtures, doesn't it? It's kind of mixtures of both. Eventually, we'll talk a little bit about this. Over time, we'll come back to this. You eventually get, if there's selection against these hybrids, and this is what we call hybrids occur in here, if selection is against them, then you kind of get blues favored here, greens favored here, and presumably you can get speciation. Okay. Sympatric speciation is the hardest. It's really hard to get. And what are we saying occurs here? Here's the original population. What happens inside that entire geographic range? Yeah, a mutation arises for a particular phenotype or something, or a particular genotype leading to a phenotype. And certain parts of it, they're what's favored, kind of the greenish one, okay? And eventually you might get to this point where you just have this big mosaic of little patches of blue and green and all this sort of stuff. You never see this in nature though. So, but the idea here is, why do you think this is so hard? Why is it so hard for these kind of green populations to arise and remain isolated and persist? What's the thing they really have to? Yeah. Well, let's just say that, in fact, that green gene is favored in that little spot right in there, right? And it really does, there's strong selection favoring it. What do you think they're all surrounded, though? Let's imagine the mutation arose right there in that little subpopulation, right, initially. What do you think what makes that really hard to get that to? Yeah, but what about gene flow? Yeah, so let's say the blue has really high fitness, blue phenotype has really high fitness in the blue parts, right? And the green has higher fitness than blues in the green parts. So let's just make that assumption. The problem is that there always is a great potential for a lot of blue individuals to go in and mate, and there's a lot of gene flow between those blue populations and that new green population. And gene flow remember what gene flow does? It kind of makes everything the same. So the problem here is there any gene flow over here? No. So you'd have to worry about gene flow, preventing the divergence. Here, there's lots of potential for it, and it makes it really hard for the process to keep going, okay? So the idea here is sympatric speciation is really, really, it's believed to be the rarest because you got all this gene flow coming in and preventing the divergence from occurring, because gene flow kind of homogenizes populations, remember? So it makes it really hard, okay? Well, let's go through some examples of this, though. This is the classic example, snapping shrimp speciation via vicariance. And this occurs not only in snapping shrimp, but 10 million years ago, was there a land bridge between Mexico and South America? Nope. There was a lot of, so there was no barrier between South America and southern Mexico. What eventually happened over time? What eventually formed? The land bridge between the two and the Isthmus of Panama, basically, until we built the canal, okay? Basically, there was 10 million years ago, these populations in the Pacific and the Caribbean, were they isolated from one another? Not really. They could move between them. Then it started to build, and about 3 million years ago, the Isthmus of Panama closed, and it isolated these Caribbean and Pacific shrimp. So you now have sort of, these represent kind of, I'm not sure what those represent, but probably water flow or water currents and things like that. But obviously, now you had a lot of water currents and potential movement of snapping shrimp between the Caribbean and the Pacific, less and less here, and now you have none at all, okay? So by 3 million years ago, the Pacific and Caribbean shrimp were isolated, okay? What do you think happened? Yeah, they became two different species. In fact, it occurred in a lot more different groups than just one. This is in your book. Turns out there are a bunch of different snapping shrimp species, and there are actually what are 6 pairs of sister species that are kind of, so here's an example, P6 and C6. This is kind of probably, they arose from a common ancestor that probably was, what, across both regions, right? When it was isolated, they became divergent. One became isolated in the Pacific, one became isolated in the Caribbean. Here's another example of that, a Pacific and a Caribbean isolate. We're not going to go into all the details of these other things. But what you have are these 6 pairs of sister species from opposite sides of the isthmus, okay? And so basically what you've got in probably about 3 million years, the formation of a bunch of new species due to this vicariance event, okay? What would you want to know if they were really truly different species? Yeah, can they mate with one another? Obviously, there's no potential for them to do naturally, right, unless they can get through the Panama Canal, which they can't, okay? But there's no potential, but you might want to see if they're reproductively isolated and how well their offspring do when you mate them, when you do hybrid matings, and we'll come back to that, okay? I'm not going to, do I really want, nah, I don't want to, you don't want to know all this stuff, do you? Nah, you don't want to know all this stuff. Anyway, I'll just tell you, turns out not all of them diverged 3 million years ago. Some of them have been isolated. Some of these sister species have been isolated for over 70 million years. And they're probably ones way out in the Pacific and way out in the Caribbean, right? So, and some of these other ones that diverged more recently are kind of these sort of inland coastal things. So anyway, you don't need to know that. Okay. So this is kind of another example. This is kind of a classic example of how divergence occurs in adaptation to different habitats. And this is not in your book. It's from a previous textbook that I used, and I don't like the examples that you use in your textbook, so I included these. So some really important mechanisms of divergence between populations, presumably leading to new species, is adaptations driven by natural selection to different kinds of habitats. And in this particular case, particular kinds of environments. So there's still some debate about this, but these are monkey flowers. And these are often called ecotypes. Some people now want to call them unique species in California, but that still hasn't been determined yet. So here's what the monkey flowers look like that live in the coastal California along kind of along the northern coast of California, and along there it tends to be kind of moist, wet, those sorts of things. This is what they look like there, and that's what the monkey flower looks like inland. Do those look different to you? Yes, those are very different. Okay, so here you have a lot of leaf material, very limited level of, very few leaves in the inland type, okay? Turns out these coastal plants that live along the coast of California, that's much moister, wetter, and those sorts of things. In Northern California, these are perennials. That means they live 2 years. These inland plants, they're on the other side of the coastal mountain range, or even near the Sierra Nevadas. It's extremely dry there, and they don't have much precipitation, especially certain times of the year. These inland plants are annuals. Okay. So how would you test whether these are, in fact, well adapted to the particular environments in which you find them? So here's how we characterize it. Coastal, the ones that live along the coast, tend to live in moist, there's more precipitation, moister environments. It's a foggier environment. There's a lot more rain in terms of the year. They tend to be very moister, wetter environments, and they flower in late summer. And as I mentioned, they live 2 years. They're perennial plants, okay? So they flower in late summer. Inland populations are extremely hot, dry summers, and they flower and set seeds in early summer and then die. They're annuals. So they don't live. They germinate, produce flowers, die, all in the same season, and they do it very early on. So how would you test whether they're well adapted, and they've sort of adapted to these unique environments to become phenotypically divergent, and would be the way to do that? Well, what you'd want to do is what's called a common garden experiment, where you put plants from each population, right, into each environment and see how they do. So what do you think? Who's going to do best in the coastal field site? And I'm not going to worry about this gene insertion. Don't worry about that. Who do you think is going to produce more flowers and have a higher fitness in the coastal field site? The annual or the perennial? So I think what you would expect, so what's shown here is this is the proportion that flowered when you took plants from both populations and you planted them into the coastal field site. Here's the proportion that flowered that were inland, the inland annual plants. Here's the coastal perennial plant. We're going to ignore these other two major lines. Which one produced more flowers? Yeah, the perennial, the coastal plants, okay? And we don't have to worry about the fitness, but what's the fitness of the inland plants in the coastal site? It is really low. They don't do well at all. So not only do they not flower, they actually don't live very long. Which one has a higher fitness? The coastal one. So all this says is, in this particular site, the coastal plants do best in their original environment. When the inland plants don't do too well. So what do you expect to happen in the inland field site? Just the opposite, right? So here's the inland, the real dry, hot, dry summers. Who does best there? Look at that. It's really different there. Almost 90% of the inland plants flowered. How did those bushy, flowery, the ones with a lot of leaves, how did they do? They basically died. They had all these leaves and wanted to produce a lot of leaves, and they had no water, right? Guess what? They died. So really strong selection against the coastal plants in the inland site, and their fitness is pretty much zero, okay? So basically all this shows is these things are extremely well adapted to the environments in which they live, and they have particular kinds of phenotypic traits that maximize their fitness in their particular environments, okay? Do you think they're different species? Come on, I do. Say they are. Okay. Okay. Okay, here's the best example of sympatric speciation, and this has been replicated in about, I don't know how many species pairs in these African cichlids. Okay, so I put a question mark here. There's strong evidence that there's been, and I guess it depends on how you define sympatric. We're not going to worry about it, but Lake Victoria cichlid males, particular sister species of these cichlids, when you look at two species that are really closely related to one another, the way that they differ is not so much in terms of what they eat, what their body shape looks like, but is really their male coloration, okay? And so Lake Victoria cichlid males often differ in coloration. So it's kind of hard to see, but these two species right here are sister species. They're the two most they're closely related to one another out of the hundreds of species in these Lake Victoria cichlids. Okay, here's the red male, and this is kind of the bluish male. Okay, so the idea here, what they think happened, and they think this happened a lot, when you go to these rift lakes in East Africa, whether it's Lake Tanganyika or Lake Victoria, this is what you see a lot of. They eat a lot of different things, but often a lot of the taxa, the species pairs differ in their male coloration. And they kind of live in different parts of the lake. Some kind of live on the shoreline close to the top of the water. Some live much deeper in the lake, okay? And it turns out, guess what happens to sunlight as it hits water? Certain parts get absorbed, and then certain wavelengths transmit deeper into the water, okay? So if the water is clear, different habitats influence the traits. Okay, so The idea here is that red wavelengths, in general, penetrate deeper into the water. And blue wavelengths are absorbed at the surface. So you have a sort of blue wavelengths that are primarily at the surface, and then these red wavelengths go deeper into the water column in the lakes. Okay? Where do you think these two males are found? Huh? Some are in deep water, some are in shallow. Okay, I'm going to tell you, one of them is in deep water, and one of them is in shallow water. Which one? So blue wavelengths are absorbed at the surface. That is, they're kind of present in the surface, right? Where do you think this one's found? In the top. Red wavelengths penetrate deeper in the water, and it turns out in all these taxa, all of the red males, red species, are found deeper in the water column, down at the bottom of the lake. These often tend to be either swimming up in the upper water column or very close to the shore in kind of shallow waters. Okay. So the idea here is these different habitats influence these traits involved in, we're going to talk about a little bit about how female preference and sexual selection actually drove this, that these populations diverge in mating preference, and these opsin genes, I'm going to show you the results. So there are particular kinds of genes, these are photopigment, these are in vertebrate retina, right? You have these opsin genes that get activated or excited or turned on by different wavelengths of light that we use for sort of color perception, right? And there are 4 major opsin genes, and they kind of differ in terms of which wavelengths of light they respond to. So this is one of the most complicated graphs I'll ever show you. So you need to understand this in great detail. No, I'm just kidding. Okay. So the idea here is what happened. They actually think this is what's called sensory drive, or kind of like the Tungara frog example, where it leads to strong female preference, and you only find these distinct male phenotypes in clear water. Do you know what's happening in a lot of these rift lakes due to agriculture and runoff and things like that? Guess what's happening to a lot of these lakes in certain parts of them? Actually, they're getting kind of murky. They're getting kind of muddy, okay? So I'm just going to show you, we're going to kind of look at what happened over here. Let's look at this. This is a site. These are 5 different sites within Lake Victoria. So we're just going to kind of look at the extreme, okay? This is the one that has extremely clear waters, okay? So here is the sort of coloration of the males as a function of the depth of the water. So here's near the surface, right? Here's 1 meter below the surface. Here's right at the surface. And here's much deeper water. What do you find there? In this really clear water, where's the blue species found? Up at the top of the water column, right? What's in the much deeper part of the water? The red males. Okay. Is there a clear distinction between those two? Yes. Here are the reds below that depth. All the blues are above that depth. So there's a clear vertical separation of these two species. And it's always the male coloration. So they're always the blue males up in here, the red males down at the bottom. Okay. I'm not going to go into worry too much about all the details here in terms of these genes, but it turns out that these are the genes for the opsin genes. And it turns out all of these red males down in here, basically the females have these opsin genes that are most stimulated by red coloration. This is the other pie over in here. These are the females over here that have these sort of blue genes that are the blue opsin genes that are most stimulated by blue, and they tend to be most common up in here. And it turns out when you give these females from these different populations, if you take a female and give her a choice, and she's from up in here, this species, guess what she prefers? Which kind of males? She always prefers the blue males. You take a female from this population down in here and give her a choice between a red and a blue male, guess who she prefers to mate with? Her red males. So there's real strong female preference for the particular coloration that she associates with. Okay. So the idea here is there's strong female preference for the coloration, and it's the signal that's most readily visible to them. Blue's really visible here, red's really visible down there, and that's what she chooses. And the idea here is, I would call this sympatric speciation. These things are literally living right on top of one another, right? These things, you know, that's not a big difference, right? You're talking about 1 meter. They're right next to one another, and they've evolved these different colorations. So what do you think happens? I'm not going to go into all the details, but guess what happens over here? So these are all populations that the water has become murkier, less clear. It's mostly from agricultural runoff, streams running into the lakes, or algal blooms, all sorts of weird things are happening in some of these lakes. Has something happened? Females cannot distinguish red and blue males anymore in that light environment. Those signals are lost. Guess what's happening? They're all mating with each other, and it's all getting screwed up, okay? So there's basically these females, you give them a choice, they just kind of mate at random, okay? I'm not going to go into all the details, but what's happening, what do you think, I don't know if you're concerned, should we be concerned about that? Maybe. I mean, you might say, who cares? But anyway, I kind of care. So what's happening is this sort of change in the water clarity is reversing the evolutionary process, right? It's basically causing what were once distinct species, right, to what? Become the same. And there's a lot of hybridization between these things, and people are worried that there's going to be a decline in the species diversity in these African lakes because of this. So what they're really trying to do, and a lot of the East African nations are trying to control the agricultural runoff, the nutrients, and all these sorts of things to make sure that the water clarity. Yeah.
So in that environment, you said that only females meet with red and blue.
Yeah.
So you can't really call that a different species though, because they can still mate in other
Okay.
environments.
Okay, so she said we really wouldn't consider those different species because they can mate with individuals in a different environment. Would anybody want to argue against that?
Isn't it at the point where they don't know yet when they will evolve because it's still too early?
Well, okay, I'll argue against both of you. Okay, what I would say is that what you're showing from this example is that females in that particular area, right, show an extremely strong preference for the red coloration. Huh? Yeah. Yeah. The option genes that they have are triggered by different wavelengths of light. And what it leads to is strong preference for that particular male coloration in that environment, right? And a strong preference for a different male coloration in that light environment. Okay? And the idea would be that potentially this has led to it's a little different than the complete vicariance isolation, but you still get this isolation on this small spatial scale where selection for blue males, selection for red males. Yeah.
So are they survival offspring?
Yep. So what they haven't done is what you would really want to do is what you would want it. So what would be the next thing you want to do? And I might leave it at that. Okay? Would you like to try to mate a female from here with a male from here and see what those offspring look like?
Isn't that what happened in
Yep. Yep. That's what. Yep. And that's kind of what they did in this particular experiment. And what it turns out is those hybrid offspring, right? And that's what was kind of shown. Guess what that is? That's the hybrid offspring between these two. Guess how these things do? They don't do well at all. Well, they don't do well in terms of females. If you give this female a choice between her own male and that male, guess which one she wants to mate with? Her own. Okay. So there's really strong. There's a strong indication that there's strong selection against those hybrid offspring, and that's the next. So I think I'm going to leave. So And this kind of leads nicely to the next thing, which is what we call the potential for, barriers can break down, right? Things can become divergent. Then a barrier can break down, which is kind of what happened in the unclear waters. And then you get hybrid matings. So we're going to then talk about what are the consequences when two isolated populations now come back into contact and what happens. It can often reinforce the speciation process. So we'll be talking about secondary reinforcement. Okay. And then we'll finish that, and we'll try to get through the phylogenetic tree stuff. Okay. Oh, let's do an eye clicker. I'm sorry. Just say, yeah, I'm not doing any questions. I forgot, I didn't have time. Just hit anything. Okay.